I'm not sure. It was me inhaling 3 eggs as I walked over here. I'm not sure if that qualifies as breakfast. But all right, so I'm going to write down here. Or maybe I should try to write left. Let's see how that goes. I got 2 colors with me. That's no good. God, I came prepared for once. Okay. We got to answer that question. All that crap we did the other day, how is it different if fertilization occurs? The whole first half is the same. If you recall this graph. Let's do the graph again. You know, this is days. And we had things like, I'll just do one of them. LH. We had LH levels going up and then like that. Doing that. We had estrogen levels also going up, and we said if that happens, if the estrogen levels go back down again, that low level of, I shouldn't say estrogen, I should say sex steroids, because it's really both estrogen and progesterone. If those levels of sex steroids are going to drop like that, then, there we go, like that. Then you're going to wind up back at day 0 of the next ovarian cycle. Uterine lining gets shed because it can't be maintained because the levels of sex steroids aren't high enough to maintain it. So what's going to happen then if fertilization of the egg cell occurs? Because if it does, you better not shed a uterine lining because then there's no place for the embryo to implant. So let's go back and look at the event fertilization. You know a little bit of this from last semester. We did some early animal embryology, so some of these terms are familiar already. But if we go back to looking at the events of fertilization quickly and then say, all right, then what? First of all, it's important to note that fertilization does not occur in the uterus. It does not occur in the ovary. It occurs in the fallopian tube. So if you recall, right at ovulation, right here, an egg cell, here's an ovary, right? An oocyte, and a few of its associated granulosa cells got released into a fallopian tube. Here's a fallopian tube. Here's a uterus down here. There's another fallopian tube. And right now, the lining of the uterus, you know, we're right about here. The lining of the uterus is significantly thick because estrogen and now progesterone levels are significantly high. So that's all good right here. Okay. So first of all, what's moving that embryo, that oocyte down the fallopian tube? There are cilia in the lining of the fallopian tube that kind of push it along. So it's got the ability to travel like this. It's going to take a while, but it can travel. So if sperm cells show up in the fallopian tube, then you can get a fertilization event to happen. Let me enlarge this picture a little bit. Here's the oocyte. Here's some of the granulosa cells that kind of went along for the ride, and we'll get some sperm cells showing up here. There's a sperm cell. Okay. There's others. So, a few things that sperm cell is encountering as it gets up the fallopian tube. First of all, it had a, considering it's a really small cell, it had a really long trip. So, of the 500, 600 million or so sperm cells on average that land in the female reproductive tract per average ejaculation, you're not going to get very many of them that are going to last long enough to get to the fallopian tube. Maybe 100 or so of them. Not 100 million, I mean like 100. So if you're a sperm cell, the odds that you actually make it there are extremely low. But that's a built-in mechanism so that the most vigorous, the most presumably genetically vigorous sperm cells actually are the ones who have their DNA passed on to the next generation. So that's intentional. So as the sperm cells are sort of swimming their way up here, they have to, they're going to be going through some sort of hostile environments. First of all, sperm cells prefer a slightly basic pH, and the pH in the female reproductive tract is about 5.5, which of course is acidic. The sperm cells bring with them buffers. So as we will see on Friday, or actually we'll do it in lab this week. As we'll see in lab this week, one of the components of seminal fluid is some pH buffers because you need to buffer that acidic pH. So they're okay in that department. Another thing that happens, those sperm cells, when they leave the male, are actually not competent at fertilizing an egg cell. They have to be exposed to chemical factors specific to the female reproductive tract so that they can undergo changes in their cell membranes such that they can actually fertilize an egg cell. That process, those changes that they experience as they travel through the female reproductive tract is collectively known as capacitation. So capacitation is the collectively is the changes that the sperm cell experiences as it swims through the female reproductive tract. If it doesn't get capacitized, that's a real word, if it doesn't get capacitized, it can't fertilize an egg cell. So it doesn't really look any different under the microscope, but it is different. Okay. When they do in vitro fertilization, they have to artificially capacitize the sperm cells, otherwise they won't do their job. They have to trick them into thinking they're in the female reproductive tract, which is just a matter of chemical environments. Okay, so let's assume that sperm cell and 99 others wind up in this general vicinity. So here's okay. First of all, it's really much, much smaller relative to the egg than what I've drawn, so don't take that too literally. But if you're that sperm cell, you've got a couple of barriers in front of you. Your goal is to take your DNA and get it into the cytoplasm of that egg cell so that the DNA can align and you have a zygote. But you got some barriers in front of you. First of all, you've got this wall of granulosa cells, and second of all, you've got this layer, I will highlight it in blue, you've got this layer of material before you can even get to the egg cell's membrane. That material that I drew in blue there, it's kind of a coating around the egg cell. It's made of some protein and some polysaccharide. It's called the zona pellucida. So the zona pellucida is this material, it's this coating, this layer of, it's a combination of some proteins and some polysaccharides, mostly protein, that coats the egg cell. If you're the sperm cell, that's just another thing you got to get through. So you've got some challenges here. This layer of granulosa cells, remember this is really three-dimensional. Imagine this as like a sphere here. This layer of granulosa cells is something you have to squeeze your way through also. That layer has a name, of course, everything has a name. It's called the corona radiata. The corona as in crown. So somebody thought it looked like I don't know who makes this crap up. It's a crown of cells around the egg. I don't know. It's whatever. They had nothing better to do when they made up these names. Anyway, so that sperm cell and others that made it this far are going to squeeze their way through these cells, make your way through there, and here you are after you squeeze your way through there. And you're going to have to actually bind to and fuse to that zona pellucida. And this has to be specific. You want to make sure that there's a crosstalk, or that there's recognition between the egg cell and the sperm cell. You want to be sure that the membrane of the sperm cell can recognize that zona pellucida as, oh yeah, that's really an egg cell. Now I'm in the right place. You don't want to just be like running into a wall and saying, okay, here I am. So there's got to be interaction. So guess what you have? You have receptors. There are receptors on the sperm cell's membrane for zona pellucida proteins. So there are specific proteins in the zona pellucida of this egg, and they're actually named ZP1, ZP2, ZP3. It's for zona pellucida. Very creative names. But anyway, there are proteins there that are unique to, say, human zona pellucida proteins, and the human sperm cell has receptors, and it says, oh, there we go, that fits, now I'm going to go ahead and attempt to fertilize this egg. If you don't first get that, if you don't first get that zona pellucida protein to receptor reaction, the sperm doesn't do anything, and it's not supposed to. You want to make sure that, hey, that's the right egg, that's the right species, this is the right place, now I'm supposed to go ahead and do my job. So there's that interaction. By the way, I don't know if this company ever went any further, but I used to own stock in this company and I sold it and it never went anywhere, and I've lost money on it. It was called Zonagen. You can look, ZONA was the code, I don't know if it still exists even. Zonagen was developing a contraceptive that would block zona pellucida receptors on male sperm cells. So it would be an oral contraceptive for men. They would take, the receptors would be blocked so that the sperm were otherwise viable, but they wouldn't be able to bind the zona pellucida proteins, and therefore they wouldn't be able to do their job. I don't know whatever happened with the research on that. I never followed up on it. So.
Is that like male birth control?
Yeah, it would be an oral contraceptive for men. That was their goal, was to, they had it in, I don't even know if they made it to clinical trials. They were based in India, so I don't know, the research that was published was in journals. You can Google them and see if they exist. Anyway, zona pellucida. So let's say this does work. So you get some sperm cells, and not just one, but you get a bunch of them. The corona radiata is still there. I'm just going to erase it to get it out of the way. So you got a number of them showing up here. And they're all going to bind and say, hey, yeah, this is right. And of course, there's competition here. Each of these sperm cells wants to be the one whose DNA gets passed on to the next generation. So in order to get through that zona pellucida, now that you've bound to it and recognized it, you got to get through it. And then you got to get to the membrane that's underneath there and fuse with it and put your DNA in the cytoplasm of the egg. That means you got to carry some enzymes with you. If you look in detail at this sperm cell, got the flagellum, of course, but along the steps of spermatogenesis, you did a lot of throwing out of the stuff you didn't need. You know, if you're a sperm cell, once you're completed, you're only going to live a few days. So you don't need a whole lot of cytoplasm. You don't need all kinds of organelles and stuff going on in there. You carry with you some mitochondria, because you need energy. You carry your DNA that you have packaged up, and now your nucleus takes up most of you. So here's your nucleus. You streamline your shape so you can swim better, and then you carry a big old packet of enzymes. It's really just a modified lysosome. It's called an acrosome. So the guts of a sperm cell is really DNA and acrosome. And yeah, throw in a little bit of cytoplasm and some mitochondria. But it's primarily a packet of DNA and acrosomes, enzymes. Those enzymes are going to be released once you bind to the zona pellucida protein. Once you recognize that you're in the right place, it's called an acrosome reaction. You get to the right place and you say, okay, now dump the enzymes in front of me. And if all these, 100 or so sperm cells do that, there's enough enzyme to bore a hole through the zona pellucida and get to the membrane underneath. And once you get to the membrane underneath, you can bind to it. Your membrane can fuse with the egg cell's membrane, and your cytoplasm becomes continuous with the egg cell cytoplasm. Your DNA is now inside the egg cell. So that's only supposed to happen for one of these sperm cells. You got 100 or so of them hanging out here, but one of them's going to be there first. And it turns out that the instant that the first one gets through, the egg cell's membrane becomes refractory to any more. In other words, the door's shut. Because it wouldn't make sense if you had 2 or 3 or 4 sperm cells throwing their DNA, because then which one is you got too many sets of DNA in there. So as soon as the first one gets its DNA deposited into the egg cell, instantaneous change in the egg cell's membrane occurs. This is all calcium reactions and things if you look at the details. Membrane changes, and none of the rest of them are out in cold there. The rest of them are shut out. And now you have a zygote. It takes, for a human, somewhere around 6 hours or so for the sperm cell DNA to align properly with the egg cell DNA to officially call that a zygote. And that little window of time where all that's got to be sorted out. Okay. Makes sense? Everybody remembers the term from last semester, zygote? I will write it on the board just in case. Zygote's the fertilized egg. It's the one cell. And then that one cell is going to start dividing and dividing and dividing, of course. So now you have this embryo. Yes?
So how are twins?
Identical or fraternal? Identical twins is going to happen a little bit later on. So after the zygote forms, you have one cell, and if the splitting along the, we're going to look at the next two stages in just a second. If there's a fracture, let's say, in the embryo, when it's still early on enough so that the cells are non-specific, you're going to wind up with two embryos that are genetically identical. Instead of it being one glob of embryonic cells, now you got two globs of embryonic cells that then develop into two individuals, but they're genetically the same, so they're identical. If it's fraternal twins, that just means two eggs got ovulated and got fertilized, and it's the equivalent of two separate birthing events. It just happens to be simultaneous. Okay, good. So here's your zygote. This is all still in the fallopian tube, by the way. And so this new embryo, let's just draw it as a blue circle, is getting pushed down the fallopian tube, and then it becomes 2 cells, and then it becomes 4 cells, and then it becomes 8 cells, and it's still called a zygote, that embryo. Once it goes past the 8-cell stage into this glob of cells that is 16, it is called a morula. There you go. And then that solid ball of cells is going to divide and divide and divide and divide and divide. And eventually that solid ball of cells is going to sort of carve out a space inside of itself. It'll become a hollow ball of cells. That hollow ball of cells is an embryo known as a blastula. All right. Flashbacks to Bio 105. So the blastula, you will sometimes see the term blastocyst. Humans are just, I guess, taxonomically narcissistic. So there are people that will use the term blastocyst to specifically refer to human blastulas. The term blastula is generically applicable to all animals, and humans are animals. I'm just going to stick with blastula. But if you see the term blastocyst, it's a human blastula. That's all. If you look at a blastula, like I said, it's a hollow sphere of cells. I'll draw it as if it were cut open here. So, imagine a sphere and we slice it, there's the hollow space. But if you look, because we are placental mammals, there's going to be a feature here that you are going to not see in non-placental mammals. You're going to notice that there's this glob of stuff in there. In other words, even at the blastula stage, there's kind of 2 distinct sections to the blastula. Timing-wise, by the way, humans reach the blastula stage about 3 days after fertilization, if anybody's interested. So there's 2 distinct areas to the blastula. There's the glob of cells in the middle called the inner cell mass. And then there's this, what I've drawn in blue, sort of outer shell called the chorion. The inner cell mass is what's eventually going to develop into the fetus. The chorion is what's eventually going to develop into the placenta. Now remember, this is all still in the fallopian tube. There's no placenta in your fallopian tube. But if you fast forward that, that's what those are going to become. When they talk about doing embryonic stem cell research, it's the inner cell mass of the blastula cells that are used for that, because those cells at this point in time are so non-specific that if you give them the right chemical influence, they can become pretty much any other adult human cell you want if you influence them correctly. And so, if you're doing research on, I don't know, how to grow new nerve cells for spinal cord injuries, you can get some of those cells in a dish and teach them how to be nerve cells, for example. That's why they use those cells. Anyway, this is all, that blastula is still in here. It's traveling down further and further and further. So eventually, day 4, day 5, post-fertilization, it's going to get to the uterus. It's going to look more or less like that. A bunch of rapidly dividing cells. They're going to need nutrients. They're going to need oxygen. They're going to need a place to stay. And so they will implant into the uterine lining. Let's say I'll just put B for blastula. That's that, okay? The blastula is going to find its way to the uterine lining, and it's going to say, hey, this is a good place. There's lots of nutrients, lots of blood vessels, great. It actually makes some enzymes that help it sort of bore a hole and really dig into the uterine lining. That's implantation, and it's going to take up residence. That is all contingent on the uterine lining still being intact. So let's say ovulation happened at day 14, maybe fertilization happened a couple days later, who knows. Uterine lining is still high, still intact because sex steroid levels are still high. You want these sex steroid levels to remain high. If they don't, if this happens, then that uterine lining is going to disappear real quick, and that embryo is not going to stay viable. You want that to happen, not that. So that's got to look different than what we did the other day. So how are you going to make that happen? Well, during this period of time, based on what we did the other day, what structure was secreting all the sex steroids that eventually fizzled out? The granulosa cells, specifically the structure that they comprised at that point in time was called the what?
Ovary.
It's in the ovary. Corpus luteum. Remember the corpus luteum? So it's the granulosa cells that were part of the follicle, but the follicle, grafting follicle ovulated, now you get this collapsed glob of cell. They're still secreting sex steroids for a while, called corpus luteum. But if no fertilization occurred, the corpus luteum just sort of fizzles and falls apart and regresses, and then you don't have more sex steroids. The corpus luteum turns into the corpus albicans. Right? So you don't want that to happen this time, right? Because you don't want to shed the uterine lining. And if you do this with sex steroid levels, you're going to shed the uterine lining. You've got to keep the sex steroid levels high. In other words, you got to keep the corpus luteum active. Don't let it turn into a corpus albicans. Just keep, their cells are already making it. Make sure they don't regress. What was telling the corpus luteum cells to secrete the sex steroids back here? LH. But LH levels are going to drop. They're going to do what you see in blue, which means you need to find a replacement for the LH. You need to find another chemical, another hormone, that will do the job of LH, but it's not LH. You need to find somebody else that can tell the corpus luteum, yeah, stay active, even though you don't see the LH anymore, stay active anyway. What structure do you think is going to secrete a hormone like that? Who has a vested interest in maintaining the uterine lining? Of everything you see on the board, which structure is the one that really, really, really needs that uterine lining to stay intact? The what? The blastula. So guess who secretes the hormone that's going to tell the corpus luteum to stay active? The blastula. The embryo, at 3 days old, they're already making a hormone. The embryo, specifically the chorion cells of the embryo, even before they make it to the uterus, start secreting a hormone called HCG, lowercase h, capital CG. This hormone is going to overlap the function of LH. It's going to tell the corpus luteum, hey, stay active, because I'm on the way here. I want there to be a uterine lining when I get there. The way I do that is I tell the corpus luteum, don't regress into a corpus albicans. Keep doing what you're doing. And if I do that, I call ahead, make sure that this is still going on, so that when I land here, there's a nice uterine lining to attach to, as opposed to a shed uterine lining that you can't attach to. HCG stands for human chorionic. It's a tropic hormone that's talking to an ovary, right? So guess what G stands for? Gonadotropin. And pretty much the one and only time you need human chorionic gonadotropin from an individual standpoint is, well, when you're a blastula, because, you know, you got to keep that uterine lining. I'm make sure that uterine lining is intact. So that's pretty much the only time you're going to secrete it. And you secrete it for a while. Actually, the production of HCG goes on for something like 7 to 10 weeks or something like that, so that the uterine lining is maintained. If you're wondering, well, how does it get maintained after that? Because pregnancy is 38 weeks long. I'll tell you that in a second. But so you're making this HCG early on, you know, day 3, day 4 after fertilization, something like that. Since that's pretty much the only time when human cells would normally, there's pathological exceptions to this, but normally it's the only time that human cells are going to make HCG. Guess what HCG can be used for clinically? Pregnancy test. That's what they test for when they do a pregnancy test, because you either have it in your body because there's a blastula in you, or you don't because there's not. It doesn't matter how much you have. You can't be a little bit pregnant. It's either or you aren't. It's pretty much binary, you know. It's like, so you don't have to test levels. You just have to test for presence or absence. So that's the story on that.
What about false positives?
There's limits to the resolution of the test. So and there's always cross-reactivity. Most of those, certainly the home ones and a lot of the quick ones, are done based on antibodies. So if you get some cross-reactivity with the antibodies to some other protein, that's a false positive. If it's a false negative, it just means the sensitivity wasn't as good. So nothing's